This presentation will discuss the principles of emergence and reemergence of infectious diseases. The purpose of this presentation is to provide a high-level overview of these factors and concepts. We won't go into much depth or detail in the immunology, the pathophysiology, the anatomy, and so forth of these diseases. We just want to give you a broad overview of the factors and concepts as it relates to the principles of emergence and reemergence of infectious diseases. The objectives of this presentation, we're going to go ahead and discuss the terminology and definition as it's used when discussing infectious diseases, factors contributing to emergence of infectious diseases, concepts of disease transmission, and factors influencing exposure and disease. So let's go ahead and discuss some basic definitions. The National Institute of Health's Curriculum Supplement Series does a great job defining some of these terms. So the term disease refers to conditions that impair normal tissue function. For example, cystic fibrosis, arteriosclerosis, and measles are all considered diseases, but they're all very different types of diseases. For example, cystic fibrosis is basically categorized as a genetic or metabolic disease because it's caused by an impairment of a genotype. Arteriosclerosis, for example, is classified as a disease of aging because it causes, it typically becomes a problem later in life after plaques of cholesterol have built up in partially blocked arteries. And then in contrast, measles is an infectious disease because it occurs when an individual contracts an outside agent, the measles virus, which in this case. An infectious disease is a disease that is caused by the invasion of a host by agents whose activities harm the host tissues and can be transmitted to other individuals, meaning it can be infectious. Microorganisms are capable of causing disease, and these are called pathogens. And pathogens include various microorganisms such as bacteria, viruses, fungi, parasites, and so forth. The terms infection and disease are not the same. An infection results when a pathogen invades and begins growing within a host. Disease results only if and when, as a consequence of the invasion and growth of a pathogen, tissue function is impaired. Our bodies have defense mechanisms to prevent infection, and should those mechanisms fail, to prevent disease after infection occurs. Some infectious agents are easily transmitted, meaning they're very contagious, but they're not very likely to cause disease, meaning they're not very virulent. Let's go ahead and give an example. So the polio virus, it probably infects most people who contract it, but only about 5 to 10% of those infected actually develop clinical disease. Other infectious agents that are very virulent, but not terribly contagious. So the terror surrounding Ebola hemorrhagic fever is based on the virulence of the virus, which has about 50 to 90% fatality rate among those infected. However, the virus itself is not transmitted very easily. The most worrisome infectious agents are those that are both very contagious as well as very virulent. Other terms and definitions: pathogenicity. It's defined as the ability to cause disease in the host, meaning it's harming the host. Virulence, the ability to cause severe disease in the host. Outbreak. Outbreak is it's defined as a limited spread, a small cluster. Endemic. Endemic is a disease that exists permanently in a particular region or population. So, for example, malaria is a constant problem in parts of Africa. It's considered endemic there. Polio is endemic. It's still endemic in 3 different countries: Afghanistan, Pakistan, and Nigeria. Epidemic means rapid spread. So, widespread occurrence of an infectious disease in a community at a particular time. So, for example, annual influenza epidemics occur, every season. Pandemic. A disease becomes a pandemic when it spreads beyond a region to infect large numbers of people worldwide. So examples of pandemic include the Black Death, the 1918 Spanish flu. Let's go ahead and define the terms emerging infectious disease and re-emerging infectious disease. So for emerging infectious disease, it's basically defined as newly identified and previously unknown infectious agents that cause public health problems either locally or internationally. Re-emerging infectious disease is defined as infectious agents that have been known for some time, had fallen to such low levels that were no longer considered public health problems, and are now showing upwards trends in incidence or prevalence worldwide. So essentially, new re-emerging or drug-resistant infections are those whose incidence in humans has increased within the past 2 decades, or whose incidence threatens to increase in the near future. Throughout human history, new diseases have appeared periodically, sometimes wreaking substantial damage. The first recorded epidemic of the bubonic plague, for example, was in the 6th century. More recently, new diseases have emerged such as Ebola virus in 1976, HIV in the 1980s, severe acute respiratory syndrome, SARS, in the 1990s, and then the H5N1 influenza, commonly called bird flu, which first appeared in humans in 2003. These new diseases are referred to as emerging infectious diseases. Even as new diseases have emerged, some existing diseases have spread more widely in areas in which they had already been present, have spread to places in which they have not appeared before, or have taken on new forms. These diseases are referred to as re-emerging infectious diseases. In recent years, there has been outbreaks of a number of re-emerging infectious diseases, including West Nile virus in the Western Hemisphere, dengue fever, which spread from South America to the Caribbean and into the United States, and cholera in South America. So these are microbiological classifications of infectious agents. These include viruses, prion, bacteria, fungi, and parasites. And we'll go ahead and provide a brief review of the general characteristics of each of these agents in the following slides. Viruses infect all organisms, from plants and animals to fungi and bacteria. Viruses, however, are not organisms themselves, because apart from a host cell, they have no metabolism and cannot reproduce. A virus particle is composed of a viral genome of nucleic acid. Viral genomes may be double or single-stranded DNA or double or single-stranded RNA. Viruses are classified using a variety of criteria, including shape, size, and the type of genome. Examples of DNA virus are the herpes viruses that cause chickenpox, cold sores, and painful genital lesions, and the pox viruses that cause smallpox. Significant examples of RNA viruses that cause human disease include rhinoviruses that cause most common colds, measles and mumps, rotaviruses that cause gastroenteritis, and the retroviruses that cause AIDS and several types of cancers. Prions are infectious particles that consist only of a protein. So what's a protein? They're large biomolecules consisting of one or more long chains of amino acids. Proteins perform a vast array of different functions within living organisms, including catalyzing metabolic reactions, responding to stimuli, DNA replication, and so forth. During the past 2 decades, evidence has linked some degenerative disorders of the central nervous system to infectious particles that consist only of protein, and these are known as prions. So examples of some prion diseases include scrapie in sheep, and bovine spongiform encephalopathy, aka mad cow disease in cattle. And a lot of them are or majority of them, are pretty fatal. And all known prion diseases frequently result in brain tissue that is riddled with holes. So you can think of a sponge with holes in it, and that's what it does to the brain. Bacteria, they vary in pathogenicity and virulence. Some could be very pathogenic and some could be very virulent. They're characterized by a number of different categories. They're frequently divided into two broad classes based on their cell wall structures, which influences their Gram-stain reaction. Gram-negative bacteria appear pink after the staining procedure. Familiar pathogenic gram-negative organisms are Salmonella typhi, which causes typhoid fever, and Yersinia pestis, which causes plague. Gram-positive bacteria appear purple after the gram-stain procedure. Examples of pathogenic gram-positive bacteria are Staphylococcus aureus, which causes skin, respiratory, and wound infections. There are a variety of morphologies among bacteria as well, but three of the most common are Bacillus, which is rod-shaped, Coccus, which is spherical, and Spirillum, which are helical rods. Another interesting topic as we go ahead and move forward with this presentation are the resistant forms of disease, which can emerge or reemerge when bacteria, parasites, and viruses are altered through mutation, natural selection, or the exchange of genetic material among strands and species. The development of resistance is a natural phenomenon. However, it can be sped up by human inaction. It can also develop and spread faster than would be otherwise be the case because of human inaction. So for example, the failure to address it in a timely and effective ways. It only took a few years after penicillin was introduced, for example, before strands of bacteria that were susceptible to penicillin had become resistant to it. So that just kind of goes to show you that resistant forms of disease can emerge and reemerge, caused by bacteria, viruses, and so forth. Fungi. These are plant-like organisms which grow by continuous extension and branching hyphae. There are various forms of these organisms. They can exist as yeast, molds, or they can be dimorphic, meaning they can exist as both. Many fungi also infect plants and animals. Examples of diseases caused by fungi are ringworm and histoplasmosis, which is a mild to severe lung infection transmitted by bat or bird droppings. Yeast of the Candida genus are opportunistic pathogens, meaning they're waiting for the opportunity to cause disease or infection. And this includes vaginal yeast infections and thrush, which is a throat infection among people who are immunocompromised or undergoing antibiotic therapy. And together with bacteria, fungi fulfill the indispensable role of decomposers in the environment. Parasites. So we'll talk about protozoa. These lack cell walls. Because protozoa do not have cell walls, which is the outer covering, they are capable of a variety of rapid and flexible movements. Protozoa can be acquired through contaminated food or water, or the bite of an infected arthropod, such as a mosquito. Diarrheal diseases in the United States can be caused by two common protozoan parasites, Giardia lamblia and Cryptosporidium parvum. Malaria, a tropical illness that causes over 200 million cases of disease annually, is caused by a protozoan, specifically Plasmodium. So let's talk about helminths. These are parasitic worms. Helminths are simple invertebrate animals, some of which are infectious parasites. Because they're animals, their physiology is similar in some ways to ours. This makes parasitic helminth infections difficult to treat because drugs that kill helminths are frequently very toxic to humans. Many helminths have complex reproductive cycles that include multiple stages, many or all which require a host. Cystosoma, a flatworm, causes the mild disease swimmer's itch in the United States. Another species of cystosoma causes a much more serious disease, cystosomiasis, which is endemic in Africa and Latin America. Another disease due to helminth is trichinosis, caused by the roundworm. Trichinella spiralis. This infectious agent is typically ingested in improperly cooked pork from infected pigs. Early disease symptoms include vomiting, diarrhea, and fever, but later symptoms include intense muscle pain because the larvae grow and mature in those tissues. Fatal cases often show congestive heart failure and respiratory paralysis. And it's interesting to know that over 1 billion people worldwide are infected with various forms of worms. Another category of parasites are ectoparasites. These are creatures living in or on the skin or hair, and these include fleas, mites, ticks, and lice. So kind of think along the lines of Lyme disease. So let's begin to discuss some ways we can classify emerging and re-emerging infectious diseases or infectious diseases in general. So you can classify them by clinical and or anatomical characteristics. So for example, the area in which they infect the anatomical part of the body. Diarrheal diseases infecting the digestive system. They could be secretory, invasive in a sense. Those diseases infecting the respiratory system, causing upper or lower respiratory infections. Those diseases infecting the central nervous system, causing meningitis, inflammation of the meninges, and so forth. Diseases infecting the cardiovascular system, your heart system, causing vasculitis, inflammation of the blood vessels, or endocarditis, which is inflammation of the endocardium, and so forth. So these are just classification by anatomical location or clinical characteristics. You can also classify infections by their reservoir or pathogens by their reservoir. So the reservoir for a disease is the site where the infectious agent survives. So for example, humans are the reservoir for the measles virus because it does not infect other organisms. Animals often serve as reservoirs for diseases that infect humans. The major reservoir for Yersinia pestis, which is the bacteria that causes plague, is wild rodents. There are also non-living reservoirs. Soil is a reservoir for many pathogenic fungi as well as some pathogenic bacteria, such as Clostridium tetani, which causes tetanus. So you can classify them by, again, the reservoir, which can include humans, animals, the environment, and what are some examples of environmental reservoirs. That could be soil, water, fomites. Fomites are inanimate objects. It could be vector-borne, and so forth. One study examined 335 events related to emerging and re-emerging infectious diseases that occurred between 1940 and 2004. This analysis revealed that about 60% of these events were related to zoonosis, which is the spread of infection from animals to humans. The study also indicated that most of those events came from wildlife and that wildlife were related to increasing share of emergence and re-emergence infections over time. About 23% were related to vector-borne diseases, which are spread by arthropods such as mosquitoes, ticks, or fleas. The Global Outbreak Alert and Response Network, it's abbreviated as GOARN, verified 578 outbreaks in 132 countries that occurred just between 1998 and 2001. You can also classify infectious diseases or pathogens by the classification mode of transmission. So it can include type of contact, whether it's direct or indirect. Infectious agents can be transmitted through either direct or indirect contact. Direct contact occurs when an individual is infected by contact with the reservoir, for example, by touching an infected person, ingesting infected meat, or being bitten by an infected animal or insect. Transmission by direct contact also includes inhaling the infectious agent in droplets emitted by sneezing or coughing, and contracting the infectious agent through intimate sexual contact. Some diseases that are transmitted primarily by direct contact with the reservoir include ringworm, AIDS, trichinosis, and influenza. Indirect contact occurs when a pathogen can withstand the environment outside its host for a long period of time before infecting another individual. inanimate objects that are contaminated by direct contact with the reservoir. For example, a tissue used to wipe the nose of an individual who has a cold, or a toy that has been handled by a sick child, maybe the indirect contact for a susceptible individual. Ingesting food and beverages contaminated by contact with a disease reservoir is another example of disease transmission by indirect contact. The fecal-oral route of transmission, in which sewage contaminated water is used for drinking, washing, or preparing foods, is a significant form of indirect transmission, especially for gastrointestinal diseases such as cholera and rotavirus infections, as well as giardiasis. Other modes of transmission of infectious diseases include they could be vector-borne, sexually transmitted, and so forth, as well as airborne or respiratory. So these are some examples of encounters in disease prevention. So we'll go through a couple of these. So for gonorrhea, syphilis, chlamydia, and HIV, for these diseases, what type of contact can be required? It could be a sexual contact. What's the source of infection, the person? What's the strategy for prevention of this disease? Altering social behavior, and the preventative aim could be just avoiding contact. So for malaria, how can you contract malaria? The type of contact is through an insect, and the source is a mosquito. Prevention strategy could be insect control, and preventative aim is eliminating the vector altogether. Let's go ahead and briefly talk about the process of infection. So in order to survive and multiply in a host, a successful pathogen must be able to 1. Colonize the host, 2. Find a nutritionally compatible niche in the host, 3. Avoid or circumvent the host's immune system or immune responses, 4. Replicate using host resources, and 5. Exit and spread to a new host. So pathogens have evolved over time to try to exploit the biology of their host organisms to the best that they could. There are two main classifications of infections by site, and these are localized or generalized, and we'll talk about this in the next slide. Localized, as the name suggests, is just local to one area of the body. So, for example, the agent enters the body and reaches its target site. The agent adheres to or enters the host cell and multiplies at site of infection. The infection spreads within site, so for example, the respiratory tract or the intestinal tract. And at this time, symptoms can appear. Organism does not spread through the lymphatic system or reach the bloodstream. And because it does not reach the lymphatic system or the bloodstream, it doesn't go further from that site. So the infection subsides due to host defenses, for example, our immune system response, and then the agent is eliminated from the body. Generalized infection is when the agent enters the body, it adheres to or enters the host cells and multiplies at the initial site. Infection spreads through bloodstream, tissues, lymphatic system, or other routes, and this is when symptoms can start to appear. Infection spreads to other organs, tissues, and cells through the bloodstream, and this is when symptoms can become even more severe. And then agent eliminated by host defenses, our immune system fights it off, or disease continues leading to irreversible damage or even death of the host. And that's for generalized infections. Let's go ahead and talk about some factors influencing exposure and infection. So specifically, we'll talk about agent factors. What are factors that affect agent susceptibility? And when we talk about agent, what is agent? These are pathogenic agents. These are bacteria, microorganisms, bacteria, viruses, fungi, parasites, and so forth. So what are some factors that influence their ability to cause infection and disease in the host? So first includes sources, reservoirs, transport, and persistence in the environment, their ability to enter a portal in the human or other hosts, their ability to reach and proliferate at site of infection in the host, and then the ability to actually exit the host. So to go ahead and start discussing some factors as it relates to host susceptibility, and these include host genetics, immunity status, nutritional status, age of subject, and so forth. We'll discuss each one of these in a bit more detail. Host susceptibility factors dictate which people exposed to disease will actually become infected, which ones will actually develop the disease, which ones would actually resolve the infection, or die as a result of the infection. Defects of host defenses are major predisposing factors for infection. And again, we'll go through some of these. Let's talk about age susceptibility. It's an important predictor of disease predisposition. Both extremes of age, for example, the older you are or the younger you are, you have higher infectious disease incidence and case fatality rates in these extremes. And it has to do a lot with your immune system status. Gender susceptibility. Many infectious diseases are more commonly seen in one sex. Sex-specific diseases are due to differences in exposure, anatomy and physiology, culture, or currently unknown factors. So, for example, pelvic inflammatory disease is more commonly seen in women. You'll also see schistosomiasis more commonly in females because in certain cultural areas, gathering water, laundering clothes exposes women to these types of diseases more often and more commonly. Pregnancy susceptibility. Physical and hormonal changes associated with pregnancy causes mild immunosuppression that makes females more susceptible to certain infectious diseases. Genetic susceptibility. Which is a relationship between heredity and disease complex. It can result in reduced or even enhanced susceptibility to certain diseases. Immunosuppressive susceptibility, which increases your susceptibility to opportunistic infections, which are those infections waiting for the opportunity to infect. It varies with underlying cause and must exceed a threshold of predisposition. So if you're immunocompromised, you're at higher risk of contracting infectious diseases because your immune system is not optimally working to fight off pathogens that are invading it. Pre-existing or intercurrent infection susceptibility. Certain infections occur more frequently in or exclusively in hosts recovering from or currently infected with another agent. So, for example, if you ever have a cold and you're recovering from a cold and then all of a sudden you have a bacterial infection, so it could kind of they're pre-existing or intercurrent infections. So damage to mucous membranes or cilia-bearing cells of the upper respiratory tract by a viral infection increases the risk of pneumonia, which could be a bacterial infection. Poor hygiene susceptibility. It's important to overall health and health promotion for each individual in home as well as in the community. Poor hygiene is associated with disease transmission in the community and at home. So, for example, cholera outbreaks have a lot to do with poor hygiene. Nutritional susceptibility. Dietary habits influences host's risk of infection. Good nutritional status obviously is critical to maintenance of good health, and malnutrition is a major contributing cause of infectious disease-related morbidity in developing countries. Substance abuse susceptibility. IV drug users are at higher risk for bloodborne infections, and long-term drug addiction may reduce other host defense mechanisms, causing increased risk of contracting infectious diseases. Medical treatment susceptibility. So, non-adherence to antibiotic therapy is associated with emergence of resistant factors. The factors that contribute to the development of drug resistance are well known, and these can include the increasing use of drugs, poor prescribing and dispensing practices, inappropriate use of the drugs by prescribers, dispensers, and patients, failure of patients to take appropriate doses of drugs. For example, you're prescribed 90 days and you decide to only take 60 days. The use of poor quality or counterfeit drugs that do not contain the appropriate level of therapeutic ingredients. There was a recent CNN article regarding this. Too much use of antibiotics in agriculture, cattle, and poultry raising and fish farming. And then weak health systems with poor laboratory capacity to diagnose disease and test for drug susceptibility. Some of the factors that might contribute to the more rapid spread of resistant forms of disease due to medical treatment and medical practices include weak infection control in healthcare settings, poor sanitation and hygiene, and lack of surveillance leading to late detection of the disease. Now let's go ahead and talk about some of the major factors contributing to the emergence of infectious diseases. And these include human demographics and behavior, technology and industry, economic development and land use, international travel and commerce, microbial adaptation and change, breakdown of public health measures. And we'll go ahead and discuss each one of these in a bit more detail. One thing I want to kind of discuss a little bit more is just the breakdown of public health measures. It is important to highlight that weaknesses in public health measures or breakdowns in public health services can also contribute in a number of ways to the emergence and reemergence of infectious diseases, including the development and spread of drug resistance. From the end of World War II until the advent of HIV, for example, there was an increasing sense in high-income countries that infectious diseases can be conquered. As a consequence, many countries scaled back their attention to such diseases, including TB. This reduction in attention to TB was associated with the resurgence of TB and drug-resistant TB in a number of settings, such as in New York City in the late 1980s. And we'll go ahead and again talk about some more of these in more detail. So human demographics, behavior, and society. Some of the factors that are included within this category include sexual behavior, drug abuse, travel, diet, urbanization, war, and so forth. Poor populations, they're a major reservoir and source of continued transmission. Poverty, another big one, malnutrition, severe infectious disease cycle, and then lack of funding, poor prioritization of health funds, misplaced and curative rather than preventative infrastructure, failure to develop adequate health delivery systems. These are all human demographics, behavior, and societal problems that can cause emergence of infectious diseases or reemergence of infectious diseases. Migration and travel introduces infections to new populations. International travel as a result of trade and tourism contributes to global dispersion of disease agents, disease reservoirs, as well as vectors. Technology and industry, long-distance livestock transportation, irrigation, globalization of the food supply, and so forth. These are all contributing factors to emergence of infection. It is clear that change in many of these factors and change in their relationship with one another have been linked to the emergence and reemergence of infectious diseases. Change in the environment and land use, for example, can have a major impact on disease emergence. This could include the well-known example of Lyme disease in the suburban United States, as housing has pushed up against deer populations and deer ticks have spread Lyme disease to humans. The emergence of Ebola virus as populations have pushed up against tropical rainforest also shows the potential impact of environmental change on the emergence of disease. The increasing amounts of travel and commerce in food and other goods also have the potential to spread infectious diseases more rapidly than ever. Improvements in technology may yield many benefits, yet they might also create the conditions for emergence of disease, such as Legionnaire's disease in the cooling towers of air conditioners. Other environmental factors include flood, drought, global warming, deforestation, and so forth. So let's now switch gears a little bit and talk about bioterrorism. The potential exists for deliberate release of infectious agents, and as the Centers for Disease Control and Prevention put it, a bioterrorism attack is a deliberate release of viruses, bacteria, or other germs, aka agents, used to cause illness or death in people, animals, or plants. These agents are typically found in nature, but it is possible that they could be changed to increase their ability to cause disease, make them resistant to current medicines, or to increase their ability to be spread into the environment. So as per the CDC, bioterrorism agents can be separated into 3 categories depending on how easily they can be spread and the severity of illness or death they cause. Category A agents are considered the highest risk, and Category C agents are those that are considered emerging threats for disease. So for Category A, these are high priority agents, and they include organisms or toxins that pose the highest risk to the public and national security because they can be easily spread or transmitted from person to person. They result in high death rates and have the potential for major public health impact. They might cause public panic and social disruption, and they require special attention or action for public health preparedness. Category B agents, these agents are the second highest priority because they are moderately easy to spread. They result in moderate illness rates and low death rates. They require specific enhancements of CDC's laboratory capacity and enhanced disease monitoring. Category C, these are third highest priority agents, and they include emerging pathogens that could be engineered for mass spread in the future because they are easily available, they are easily produced and spread, and they have potential for high morbidity and mortality rates and major health impact. Emerging and re-emerging infectious diseases are exceptional examples of critical global health problems. They can arise anywhere and at any time. They can spread, sometimes rapidly, within and across countries. Different countries, with the help of various international organizations and networks, have to work together in technically sound ways and sometimes urgently if the problem of these diseases is to be addressed effectively. In some respects, the development of emerging and re-emerging infectious diseases, for example, including antimicrobial resistance, is inevitable, given that it partially arises as a result of natural processes. On the other hand, we do have control over some of the factors that help drive the development of these diseases. In principle, for example, population pressure on the environment could be reduced, and land use planning could limit destruction in animal habitats. In practice, however, these approaches will require substantial change in the way in which people live and will not occur unless incentives are in place and reasonable alternatives exist. There are also many ways we can more appropriately use drugs, including decreasing when drugs are not needed. Thus, even as people work both within and across countries to change some of the structural factors that drive the emergence and re-emergence of infectious disease, they can take more immediate actions to reduce these threats and the threat of antimicrobial resistance. Because of their nature, some measures to address emerging and re-emerging infectious diseases will have to be taken within nations, and others will require international action. So in conclusion, I did not mention many specific emerging and re-emerging infectious diseases, and the reason for that was intentional. We want, obviously, you to research these diseases, come up with your own list or table of emerging and re-emerging infectious diseases, and research these and figure out what are some of the causes, what are some of the contributing causes, whether it's host, whether it's the agent specific, environmental specific, and so forth, and go ahead and do some more research on this. And again, this assignment was left vague on purpose. We want to see your creativity, want to see your thought process and what you think is important, what you think is not important, and how you put together a professional report.